This is Professor Anant Pravade from Food Chemistry and Nutrition Department. So today we're going to discussing about the betalins. Betalins are mostly the class of red and yellow tyrosine derived pigment, and it is actually found in the plants where they can replace the other pigments such as anthocyanin. Okay, so let's move on the next slide. So this is what the betalin structure. Betalins represent immunoderivative of betalamic acid and are subdivided into the red violet and betacyanin and the yellow orange betacyanin. Okay, uh, this is water soluble and exists as an internal salt, that is sweeter ion, in the vacuoles of the plant cells. The general formula represents the condensation of the primary of a secondary amine and betalamic acid. So this is what the structure of the betalins, which is shown in the right side of the slide. Betalin chemistry. It is about 50% betalins have been identified. The majority have an acyclated sugar moiety. Moiety, in a sense, it gives the water molecule at the end of the product. The acid involved are sulfuric, then malonic, caffeinic, and synapic, as well as the citric. All betalins, all betacyanins are derived from two aglycones, that is betanidine and isobetanidine. The latter being the C15 epimer of betadine is being changed. Betanin is the main pigment of the red beet. It is a betanidine 50 beta glucosides. The beta xanthine have only dihydropyridine ring in common. The order structure of the feature are more variable than in betacyanin. Example of beta xanthine are natural volgaxanthine 1 and 2, also from the red beet. In this slide we are going to see betalin biosynthesis. Betalin biosynthesis starts with the DOPA by the opening of the benzene ring, followed by the cyclization to a dihydropyridine. The betalamic acid which is formed undergoes condensation with the cyclodopa to beta-cyanine or with some other amino acid to beta-xanthines. Red betalin is water soluble and it is used to color food. Its application is however limited because it hydrolytically decomposes into the colorless cyclodopa. The reaction is reversible. Okay. Betalins in 1963 and 1964, betanin and the indexanthin were first betacyanin and beta-xanthine structurally developed from the red beet and the cactus pear fruit respectively. Comparison with the anthocyanin. The anthocyanin bears huge from orange to red to the blue. The betacyanin may be subdivided into distinct yellow to the orange and the red violet structures. The extinction of coefficient of the anthocyanins are smaller than those for the betalins. The This means the higher coloring capacity of betalins is compared to the anthocyanin. While the betalins visibly change to the canary yellow tint at the alkaline pH through the release of betalamic acid, color strength and the brilliance will increase for the anthocyanin at the pH value below 3, rapidly decrease at the low acid and the neutral pH. Betalins are sensitive to the presence of metal, sulfur dioxide, light exposure, high water activity, enzymatic action, pH, elevated temperature. Process of beet color manufacture. First of all, we have to take a red beet, we have to wash it, grind it, mash it with the enzymation. then acidification is there, decation is there, then we have to remove its juice, we have to pasteurize it, then the juice or the color foodstuff is get concentrated. So this is the one procedure for the bit color extraction. Manufacturing of the beet color. Red beet juices, concentrate and powder are typically application of the coloring purpose authorized in the Europe and the North America. Usually whole unpeeled beet are processed. More than 30% color is lost by the removal of the peel. This is noteworthy because the greatest polyphenol oxidase activity, which is deleterious to the both beta-cyanine and beta-xanthines, is located in the film. Previously, the blanching present a tool to inactivate unfavorable enzymatic actions. Small beets are favored because they accumulated higher betalin concentration. So this is what the manufacturing process for the beet color, as we have saw in the previous slide too, that red beet is taken, we have to wash it, we have to acidify it, then raw juice we will get, we have to pasteurize, and whatever the concentration driven is there, that is called as, that is used as the food colorant. Beta color manufacturing, same procedure in this slide also. The other sources of betalins: amaranthine and I first amaranthine. R group is a gluconic acid like to 2 position of glucose, a great potential forecast for grain and leaf amaranth, both of nutritional purposes. Cultivated species exhibited higher color content than the wild species. Due to the high number of acyclated beta-cycline structure, a satisfactory stability was achieved in the spreader preparation. Because of the wider adaptability of the amaranth plant compared to the red beet, the farmer were proposed a new source of natural food colorant. Other source of betalins, cactus spear, is also gives the betalin color, although current pigment will range up to 15 to 18 mg per 100 gram of fruit. Okay, but new hybrid are proposing higher yield up to the 100 mg per 100 gram. Application of food system. Betalins are considered to be the most stable at the near neutral condition in the food. Hence, typically food commodities colored with the betalins include dairy products, some fruit fillings also, as well as some bakery product, as well as confectionery product too. Thank you so much.